Alright ladies, so with this video we're going to look into the cell cycle and cell division. The good news is most of this should really be review because the majority of you have probably learned mitosis multiple times in your life already, and so we'll go through that pretty quickly, but there's a little bit of extra detail that I'm going to want you to know that is pretty important about when your cells divide and kind of what controls that process. So we definitely need new cells all throughout our life, right? We need cells to grow and develop. We need cells for repair whenever we have an injury and things like that. And so our body needs to be able to generate new cells. Now, every cell is specialized, so the different parts of your body are going to have these different unique cells which have a unique structure and a unique function. And we call this differentiation, right? Really early in development, cells will differentiate into different types, and so you go from being this little ball of kind of generic nondescript cells to being this complex organism with all these different types of tissues. So for example, like I have on this slide, a neuron or nerve cell is a really different function and therefore a really different shape than a muscle cell, and so we need this differentiation all throughout our body. The other thing we have to realize is that you're going to lose cells. We lose cells constantly, and that's a natural process, particularly on your skin, or like when we did the lab and you scraped out your cheek cells. Those are dead cells that just flake off, basically, and so we need to be able to replace those cells. Therefore, we're going to see the most cell division in areas that tend to lose more cells. So think about areas that are exposed to the outside, right? Your skin. You lose skin cells all the time. That's essentially what dust is on surfaces, is people's dead skin cells. And so your skin constantly multiplying to replace those cells you lose. Also your digestive tract, right? You kind of scrape a lot of cells off your digestive tract when you eat and that food moves through your system. And so those tissues of your digestive tract also constantly replace and replenish cells that are lost. Now, prokaryotes, which we'll spend more time on later, they can just split into two. But when we talk about an organism like us, a eukaryote, we need a more complex process of stages to make our cells divide, and that's what we call the cell cycle. When a cell prepares to divide, it sort of has to meet a few requirements before it's allowed to do so. The first thing that we have to do is we have to replicate and equally divide the DNA so that when we end up with two cells, two daughter cells instead of just one cell, both of those cells will have an identical set of chromosomes. The other thing that has to happen, kind of combine these two bullet points here, is that the cell has to be big enough and has to have enough materials to support two cells. So you got to have enough cytoplasm, you got to have enough of the various organelles, you got to have enough specific proteins to support two cells and not just one if you're going to undergo cell division. Now this organized process, again referred to as the cell cycle, ensures that we meet these requirements and that we successfully make a new cell. So here's a picture of the cell cycle. A little bit of detail here that we haven't talked about yet, but the one thing that I really want you to notice about this slide is that the cell cycle itself involves M phase, and then the rest of this that I'm going to circle in green is all interphase, right? So when we are younger students, particularly before high school, but also sometimes in high school, we sort of learn, about interphase, and then we learn about mitosis, right? Prophase, metaphase, etc. What we oftentimes don't tell those younger students is that interphase is by far the longest part of this. I mean, look, we're talking about more than 3/4 of this cycle is interphase. So the vast majority of cells in your body right now are in interphase. And then a handful of them, which is still a big number since you're made of billions upon billions of cells, but a handful of cells are actually in mitosis and are actively dividing. The majority of cells, though, are all in interphase. Now also notice these check marks on here. We'll come back to those guys here in a minute and talk about what those check marks are supposed to be. Alright, so here is the steps of the cell cycle. Again, interphase is all of the time between cell divisions, and interphase is the longest thing, right? So again, most cells are found in interphase. We break interphase down into 3 different stages itself. The first stage, G1, which we often think of as the first growth stage, is where a cell grows. Like it says here, it makes proteins, other things like that. This is sort of your everyday, normal life of the cell. Now, from G1, a cell can also enter something called G0, or sometimes called G0 stage. Now, G0 is a way for a cell to just sort of stay stagnant. A lot of times people think of G0 as a cell being dormant. That's not a good word, because a G0 cell is still very active. It's doing what it's supposed to do. So, for instance, if it's your, this is a cell in your liver, if it's in G0, it's still doing everything the liver is supposed to do. The difference is a G0 cell is no longer actively dividing. So a G0 cell is not trying to move through the cell cycle, it is just kind of hanging out like it is. And again, we think that the majority of adult cells in people are actually probably in this G0 stage. Now, if a cell doesn't go to G0, it can move from G1 into S. And movement into the S stage means that this cell is planning to divide. S stands for synthesis, and what we're synthesizing or making is DNA. So it is during this S stage of interphase that we copy the DNA, we copy the chromosomes in preparation for cell division. Then from S we would move into G2. G2 is the second growth stage, but now we are growing specifically to prepare for cell division. So in G2 we are really getting ready to go through cell division. Now after you do those steps of interphase, you go into mitosis, so we'll look at that in just a second. And when we completely finish this cycle, the cell becomes 2 identical daughter cells. So once we finish interphase and mitosis, we finish off with 2 new identical daughter cells, identical to each other and identical to the parent that we started with. So here's mitosis. All right, remember PMAT, prophase, metaphase, anaphase, telophase. So first thing, prophase. Prophase is a busy stage. This is where we're really getting ready to ramp things up. And so what I usually teach students is kind of learn what happens in the other stages first, and then you can just remember, okay, yeah, everything else happened back in prophase. All right, so first thing, the nuclear membrane breaks down. So your nucleus essentially goes away, and the genetic material is now free in the cell. The DNA condenses from chromatin, which is just sort of like this bundle of DNA, into actual visible chromosomes, which we can now see with a microscope like ours. So whereas before we might have just seen a nucleus that looks like this, now we're going to see the nucleus gone and the chromosomes are condensed. And so that's why we often draw these like an X when we illustrate a cell. Now each of these X's is a pair of sister chromatids attached at a central point called a centromere. So essentially here we've got a chromatid there in red and a chromatid in blue. And then we've got this centromere here in green, which is connecting the two. And when we look at this, what we know is that the red one is identical to the blue one, because that's the copy we made back in S stage of interphase. Now, also during prophase, we start to form a mitotic spindle. So microtubules will start to organize themselves at these centrioles and form this thing that almost looks kind of like a football within the cell. And then part of the centromere, called the kinetochore, I know we got a lot of vocab going on right now, will connect each chromosome on that mitotic spindle somewhere. That way we can move it around. Second one here, metaphase, remember, midline. Right, so we don't really see a cell membrane in this picture here, but metaphase occurs when the chromosomes, or these pairs of sister chromatids, line themselves up in that midline of the cell. And we call that the metaphase plate, where that occurs. Third stage, anaphase, remember, apart. Alright, the sister chromatids will pull apart, so essentially each half of the X, the red piece will go that way, the blue piece will go that way. Alright, and again, remember, these two things, this blue and this red, are identical. So by pulling them apart to either end of the cell, we've divided our DNA equally, and each daughter cell will receive an identical copy of the genetic material. Last stage then is telophase. So for telophase, each set of chromatids reaches one end of the cell. A nuclear membrane reforms around each of those, and then as you can see here, the cytoplasm starts to separate in cytokinesis, giving us eventually then two individual cells that again are identical daughters, identical to each other. Then these cells move back into interphase, G1, and we start the whole thing over again. Now the part that you haven't heard as much about before, probably, is regulating or controlling the cell cycle. And as we do usually mention during freshman biology, it's really important to control the cell cycle and control the rates of cell division, because if that's out of control, we've got a big problem. And that big problem is something else that you're studying right now, which is cancer. So your body has several different levels of control within the cell cycle that help ensure that cells accurately replicate, so cells make good copies of themselves, but also that cells copy themselves enough, but not too much. Right? So the first thing, isn't really on this slide necessarily, but the first thing is that the cell is constantly checking to make sure that stages are being completed correctly. The last thing we want to do is make a bunch of bad copies, right? I mean, if I'm going to give you guys an assignment or test or something, and I make a bunch of copies and the copy machine smudges it and you can't read it, that's worthless, right? So a cell is similar. We don't want to make copies of the cell that have damage or have problems. That won't do your body any good. So we have ways to check for that. And the primary points at which the cell checks are called checkpoints. And it's a pretty obvious name, but that's what those check marks were on that picture a few minutes ago, is that there are certain checkpoints where the cell has to basically be checked out to make sure that it's ready to divide, or that the new one we're about to make is going to be okay. Now the first big checkpoint is called the restriction point, and that occurs between G1 and S. So before a cell can even head toward mitosis, we're going to check that cell between G1 and S. If the cell fails that G1 checkpoint, that restriction point, there's several things that could happen. Generally, it might just move into that G0 stage, where it will stop actively dividing, or in some really serious cases, the cell might be destroyed. But if a cell does pass through the restriction point, it is committed to dividing. So basically, once that cell goes into S phase, it is either going to become two cells, or we're going to get rid of that cell altogether if anything bad happens along the way. So as I mentioned there a minute ago, I got ahead of myself a little bit. There are checkpoints throughout the cell cycle where they will detect problems. You've got unique proteins, you don't have to worry about the names of those, that will check a cell, it will check its DNA, it will make sure that it has the proteins it should have, and if there are ever problems found, the cell will go into what is referred to as cell cycle arrest. Now the first thing that the cell will do is it will try to fix that, right? It will try to fix the DNA damage, it will try to make up for proteins that aren't there, but if it can't, the cell will die. And this is a really controlled process called apoptosis. It's a really fun name. All right, so apoptosis is controlled cell death. And a lot of times, what I like to think of this as is sort of like a clean process, right? This one single cell will die. It won't damage other cells around it, and it won't, be a real detriment to the larger organism. We're just removing a cell that we don't need. One thing that's kind of cool is sometimes apoptosis is done on purpose. So for instance, if you've ever heard before that, when a human develops, and a lot of other mammals too, your hand starts as something that looks more like a mitten, right? And then the tissue has to be removed between your fingers. Some children are born, especially if they're born premature, they're still going to have webbed fingers or webbed toes, and that's because it hasn't gone away. Well, the way that tissue goes away is controlled apoptosis, where the body is intentionally doing that. But again, if something goes wrong during cell division, the body might cause apoptosis and get rid of cells that are going to be a problem. Okay, so the question becomes, how does this work? And I'm not going to expect you guys to know a whole lot because it's sort of a tricky process, but I do want you to know some. And the way a checkpoint works is through things called cyclins and CDKs, or cyclin-dependent kinases. So, first, all right, you have in your cell, I'll draw a little graph sort of, you have things called cyclins. Now, cyclins are proteins which fluctuate their levels throughout the cell cycle. Hence, that's why they're called a cyclin, right? They accumulate and then they go away, and then they accumulate and then they go away. And this occurs at certain points during the cell cycle. Now, you also have a second molecule here called a cyclin-dependent kinase, or CDK. And this is an enzyme that can cause cell division to move forward, right? So a CDK gives a positive signal to the cell cycle. However, these are cyclin-dependent kinases. They only work if they can bind to cyclins. So a CDK, let me go back up to my picture here. All right, so a cyclin, here in red, right, fluctuates throughout the cell cycle. A CDK, here in green, has a constant concentration, a steady concentration throughout the cell cycle. But the only time the CDKs actually work are when there are cyclins to bind to them. And so sort of the way I drew this picture, right, let's say that a CDK only works when there is more cyclin than CDK in this graph. So the only time that the CDKs are actually positive are sort of like where I'm drawing these yellow lines. What it does is it allows the cell to stop cell division if it needs to. So basically, under normal conditions, you see something like this. The cyclins go up, the CDKs activate when they should, the cell continues through the cell cycle. But if something were wrong, right, let's say there is some DNA damage that gets detected, what the cell will do is it will lower the level of cyclins. Oh, I don't know why it's not all erasing. It will lower the amount of cyclins. All right, let's try something else. There we go. So to lower that level of cyclins, and then you don't get active CDKs, and so you don't get cell division. Now, what you can also look at, and I believe one of your unit homework questions kind of talks about this, is if CDKs and cyclins are always bound, then the cell is going to continue to divide no matter what, right? And if CDKs and cyclins for some reason never bind each other, then CDKs are never going to be active, and you won't get cell division at all. So a little bit complicated, but hopefully that'll make sense as you study it a little bit more. That takes care of the cell cycle. Now we move on to talk about what happens if the regulation breaks down, which is also known as cancer.